So what we're going to be doing is we're going to be going through and talking about the tissue that we find inside of our cardiovascular system. So that's going to be pretty important because this tissue is this life-sustaining tissue that distributes all kinds of things around the body. So to be sure, as we were talking about in lab, it is a connective tissue. And I was going to try to get my stylus to work, but it seems like the battery's kind of shot there. So we'll find out. Otherwise, I can draw with the mouse. Let's see. No, not today. Not today. Okay. Well, in any case, when we're talking about the blood, what we're going to be going through is learning about, like we said, this tissue. So it is a connective tissue, and it is very important in terms of maintaining homeostasis. So as I go through this material today, what we're going to find is I did modify this just a little bit in terms of the PowerPoint itself. I kind of streamlined it because last time we spent a lot of time doing our orientation and we got a holiday coming up, etc. So I streamlined this a little bit. It's the same content as you have, but it may have it does have fewer slides. I took out some of the wordy text slides. So I have a lot of the figures in there, like I recommended, just like the textbook. Really, the figures and the tables are where we want to focus our efforts anyway. It's where we have a lot of that core content. So it's not saying that other information isn't useful. I'm just we'll still cover it here. I just won't have all the slides where we'll go through it line by line instead. Okay. So what we find is, like we said, this is a connective tissue. It's a good example of a connective tissue because it connects almost all parts of the body. Rather than talking about bone or ligaments or something like that, this is a fluid connective tissue where we have cells in an extracellular matrix, just like we usually find for a connective tissue. So we have our red blood cells, like we saw in the microscope, our erythrocytes, our white blood cells, our leukocytes, as well as platelets. And so for the platelets, those ones, they actually have a specialized name that are called thrombocytes. And they are, like we mentioned last time, they're not whole cells, they are fragments of cells instead. Now outside of the cellular elements, we have the plasma, so that's the extracellular matrix. We have a liquid extracellular matrix. We talked about, like on the lab practical, how you might have a microscope on the lab practical and have a blood slide on there and be pointing to a particular cell, and you have to identify it as a neutrophil or a monocyte or something like that. In the past, I've also sometimes had a question on there that says, what type of tissue are you looking at? And to be sure, you can call it blood, but realize, like we said, this is one of our 4 connective tissue types, right? So this is not epithelial, not muscle, not nervous, but connective tissue. So we have that liquid extracellular matrix, which is called plasma. And we're going to talk about all of the various things that we find in the plasma. Sometimes people will discount the plasma. They're like, it's just a bunch of water that helps the cells to go around. And really the importance is on the cells. But there are so many things that are transported in the plasma. We want to make sure that we learn about those as well. So we'll come back to that. Okay, so like we said, blood helps us in many ways to maintain the stable internal environment. So the stable internal condition or stable internal environment, that is a definition that we went through in 201 as well. So what does that sound like? What is that? Yeah, so that's homeostasis, right? So homeostasis is how we have the stable internal condition, where regardless of what's going on outside of our bodies, we're trying to adjust our internal condition so that it stays relatively stable. So whether it's too hot outside or too cold in my office or whatever, the inner condition is going to be relatively stable, and the blood certainly helps with that in various ways. Okay, so this figure right here, which is actually taken from a different textbook, it does a good job of really laying out the important components of both the cellular elements that we see on the right, as well as the fluid component that we see on the left. So like I said, if you have a textbook or whatever, if you're looking at e-textbook, you'll find a table like this that kind of summarizes all the components of the blood. So it's going to be important that you know not only, like we said, the cellular elements that we see on the right, but all of these different constituents that we see on the left and what they're for. We always have this relationship between structure and function. What is it? Why is it there? What does it do? Okay? All right, so On the right side, we see cellular elements make up, of a sample of blood, about 45%. On the other side of the coin, the liquid component is about 55%. So that ratio is something also that we tend to maintain. Now, it's going to be different for different people, right? So it depends on sex, depends on body size, depends on physiological state or whatever, but it should be kind of in that range. So anywhere from 40 to 45% in terms of the cellular elements, and then 55 to 60% in terms of the fluid component, okay? If it gets off from that, there can be problems. So if, for instance, there are too many cells, that can be a problem. It makes it too thick. It has a high viscosity, it's hard to move around, etc. If it has too few cells, those cells all have a job to do, and so if we don't have enough of them, then they're not doing their job. So that is actually something that is called hematocrit. We talked about how we're going to do 3 tests in lab today. We're going to do blood typing, we're going to do a hemoglobin assessment, and we're going to do hematocrit. So that's one of the tests that you would do. As you can see at the top there, we're not going to do it from a vein like that. You'd be happy to know. We're going to take a smaller volume of blood. That's our intent. And then we are going to take that sample. We're going to centrifuge it. That will separate the cellular elements from the fluid component. And then from that, you can actually measure what's called hematocrit. So that's what we see right here. So, for instance, here's the top of our sample right through here. Where shall I get my little pen working there? Let's see. So the top of the sample is right through here. And then this is the bottom of the sample. So in order to calculate hematocrit, you basically measure to the top of our cellular elements. You measure that distance through here. Use that as your numerator, and then you put it over the height of your entire sample, and it gives you a percent. Okay? So we'll actually be doing that in lab. Okay. So when we look at, let's take a look at those substances and constituents of the plasma. So to be sure, water is a big constituent of the plasma. It makes up more than like 95% of the plasma. And it's not just for pushing the cells around. In fact, fluid levels in the body is one of the main things that we maintain homeostatically as well, right? So we want to make sure that everything's hydrated well. The intracellular fluid, when we talk about the cytoplasm, that is mostly water. So we have to make sure there's enough inside the cells, enough outside the cells, etc. If we get dehydrated, like think about your muscles or whatever, what's going to happen? How do we rehydrate them? Right? We ingest it, and then it absorbs across the body wall, across the intestinal wall usually. We'll learn about that in the digestive system. And then where does it go? It goes in the blood. Well, that's all well and good for the tissue right next to the intestine, but what about, your gastrocnemius muscle that you learned about before? What if that's dehydrated? You got to get the fluid to it as well, right? So there's this definite importance of water in the body to make sure all these different components are filled with water that they need. So, water and then ions. We know that ions are also known as electrolytes. They are charged particles that are inside of our fluids, and those have a role to play as well. So they, if we look at that list, we can see some really important ones at the beginning: sodium, potassium, calcium. Last semester, when you were talking and learning about muscles and nervous system, you started talking about things like action potentials and gradient potentials, right? So the movement of those charged particles across the membrane allows muscles to contract, allows neurons to send signals, etc. So we have to replenish those, we have to distribute those accordingly, and for the most part, that's happening in the blood. So sodium, potassium, and calcium, we know that there are important roles for those. Magnesium is also important in terms of bones, in terms of muscles, etc. Chloride helps to balance things out in various ways. Bicarbonate helps with pH balance in the body as well. We'll talk about that coming up. So those ions that we have there, again, they're not just floating around there. They are necessary, required components for the body. We get down below that, and we get to plasma proteins. So we know that proteins are organic molecules, much bigger than ions. These are floating around in the blood too, and there are 3 critical ones that we see here. And you should know, when we're listing these things right here, as we're doing it right now, you should be able to do it also on an exam. So for instance, as we were talking about, we have those exam objective sheets where it has kind of study questions for each of the chapters. You should know this from this chapter, know this, know this. If you haven't looked at those yet, like we talked about last time, you should, between now and next time. Exam objective sheets as well as the vocabulary sheets. Okay, so that you know there's a certain list of terms that you'll have to be able to define on the exam too. I can tell you, I believe, that in this chapter, one of the exam objectives that's in bold, which means that it could be an essay question, has you talking about different constituents or different substances that you can find in the plasma, being able to name them, and describing what's their importance, why are they there, okay? So a sample question might be like, name 5 substances that you could find in the plasma, and why are they important to the body? You can see we've got like, I don't know, what's that, like 12 of them right there. So you got lots of different examples that you could use. So water, different ions, you can name specific ions, and then we're talking about these plasma proteins. So you should know the names of these plasma proteins. The first one is albumin. So albumin is a plasma protein that's always found in the blood, and its main role is to help maintain osmotic balance. And when you look at osmosis there, osmosis is what? That's from way back in the beginning. Yeah? Right? So it's water. It's the diffusion of water from an area of high concentration to low concentration, often across the membrane. Okay? So it's the way that we might be able to regulate cell volume as well as the concentration inside and outside of cells, etc. So when we talk about the blood and how we can deliver water to dehydrated cells, etc., for the most part, and we'll talk about this when we get more to the blood vessel chapter, but for the most part, it's blood pressure or hydrostatic pressure, which is pushing that blood around and pushing that fluid into the tissues. Okay? But if we were doing that all the time and we didn't get anything back from the tissues, then the tissues would just continue to swell and swell and swell as we're pushing water into them. So for water balance, it has to be delivery to the tissues as well as kind of reclaiming that water from the tissues. It has to be an exchange. Whereas hydrostatic pressure, fluid pressure, blood pressure is pushing it into the tissues. It's osmotic pressure that tends to draw or suck it back from the tissues. And by having that albumin in the blood vessels, it creates almost like a molecular suction. It creates an osmotic gradient that draws the water back. The water kind of sees all of that, the albumin as well as other components of the blood in there, and it says, hey, there's a lot of you in there, and there's not a lot of me, relative scale. So I'm going to go from where there's a lot of water here to where there's not a lot of water in there, and I'm going to try to even things out. So it helps to draw the water back into the bloodstream. So that's why albumin is there. If you don't have that, you might get a little, swelling in the limbs or something like that because it's not helping to maintain that balance. Fibrinogen is the next one. So fibrinogen, if you look at the name, it might suggest to you that it relates to fibers or something like that, and that's exactly true. So these are going to be protein fibers that are very important in terms of blood clotting. So if you have a break in a blood vessel, if you, for instance, cut your hand on a sign or something like that, it could happen, right? You don't want that continually flowing out of you, right? We have hemostasis where we're trying to maintain the volume of blood that we have inside of the body, not let it leak all over the place. So clotting is a very important process that involves fibrinogen. The fibrinogen is kind of like there's this whole sequence of events that leads to the formation of a blood clot or a scab or whatever. And all these steps are necessary, but like the very last step, the very pinnacle step, is the activation of that plasma protein called fibrinogen. It's floating around all through the blood at all times, just in case, because we don't know where a break is going to happen, right? So you can't like wait until a cut happens and they're like, oh gosh, we got to make some fibrinogen, then we got to get it over there. By that time, a lot of that blood will have leaked out. So if we have it circulating all the time, then it's ready to go. But we keep it in an inactive form, a soluble form, so that it's floating in our plasma. But then as necessary, we will activate it, and it will become insoluble, right? So like in chemistry class, if it's insoluble, it will kind of come out of solution. And it will come out of solution, and it will form these protein threads on the break that will help to form a solid clot, so that it stops the leaking from that blood vessel, from that tissue, and then allows the healing process to occur underneath it. Okay? So very important. In fact, when it becomes activated, the name changes from fibrinogen, where it's in its inactive form, and it becomes what's called fibrin. We'll talk about that more towards the end of the chapter, where we talk about how this whole blood clotting cascade works. But that's the second important plasma protein. The last one, that group, are called globulins or immunoglobulins. These are antibodies. Today, as we're getting into our blood tests, we're going to be talking about the different blood types. How many people know their blood type in here? Okay, so yeah, probably over half or something like that. The morning section, and they might see this if they watch the recording. The morning section, it was probably less than 15% of them knew it. There was only a handful of them that knew their blood type. So during that lab, it was great because most everybody now discovered something new about themselves, what their blood type is. But as we go through that and we discuss the A and the B and the O and the AB blood types, we'll talk about antigens and antibodies. But basically, antibodies are these free-floating proteins that are circulating around looking to identify something that is foreign, something that should not be inside of your body. And so if it finds that and it matches up with it, it has these little binding pockets on it, kind of like enzymes have a little binding pocket that fit like a lock and key mechanism with a substrate. If it finds something that binds with it, it's going to hold on to it almost like molecular handcuffs. It's going to stick to it, and it's going to try to flag it and say, hey, there's something over here that shouldn't be here. Somebody help me with this, right? So there are antibodies that are floating around looking for these foreign substances, foreign cells, etc., just in case you've been infected with something. It could be a foreign blood cell. If somebody, right, gave you a transfusion, they didn't match up your blood type, we want to get rid of those foreign blood cells. But it could be like a bacterium or a virus or something like that. So antibodies are constantly floating around as well, those immunoglobulins. Okay. Beyond that, if we look into that little category at the bottom, it seems small, but there's a lot of important things there, right? Nutrients. When we start talking about the digestive system and digesting our carbohydrates and our lipids and our proteins, etc., we break them down to these small building blocks that we want to give to the body, give to the tissues so that they can do what they need to do. And so they're going to be circulated around via the blood. So there's going to be nutrients in there, the little building blocks from our food. Waste products from metabolism are going to be in there because we're going to try to get it to like the urinary system or something like that. Respiratory gases. We will cover respiratory system after cardiovascular system. We'll talk about how we can exchange carbon dioxide and oxygen with the lungs. But we also have to transport those gases, again, to things like your gastrocnemius that's working, or to your brain that's working, or whatever. So there's always going to be some oxygen or carbon dioxide that is going to be transported by the blood. Now, for the most part, carbon dioxide is transported in the plasma. For the most part, oxygen is transported bound to the red blood cells, to the erythrocytes. But there's a small fraction that's still floating around in the plasma by itself. So, at the very last there, we see it says hormones. So hormones are created by endocrine glands. They are chemical messengers, kind of like neurotransmitters, but rather than being transported by an axon and released onto a muscle cell or something like that, they are dumped into the blood. The blood circulates it around, and then the target tissue has receptors on the cells that then get that message, and it basically stimulates that target tissue. So, various hormones would be in the blood too. So lots of things that we can find in the plasma. Questions on that? Okay, so let's look at the other side of the coin when we look at the cellular elements there. So for the cellular elements, we have three main categories, right? So we have the red blood cells, we have the white blood cells, and then we have the platelets. And again, we keep talking about these as cellular elements or formed elements or something like that rather than cells because erythrocytes, the red blood cells, are not whole cells. We'll talk about how the fact that they give up a lot of their organelles when they mature. Leukocytes, the white blood cells, are whole cells, to be sure. They have a nucleus, we can see, they have a stain. We use the shape of the nucleus, the size of the cell, those granules to identify them, the 5 different types. But then the platelets, to be sure, not whole cells. They are just little cell fragments, in fact. Now, each of those different three, if you will, cellular element types have a role to play. Erythrocytes, red blood cells, mainly transporting oxygen. We'll talk about how they do that. The leukocytes, the white blood cells, all 5 of them help defend the body, identify what should not be there and try to get rid of it, but they do so in slightly different ways. So we'll talk about how they do that. And then the platelets are also involved in blood clotting. So like we said, it's a whole sequence of events that lead to that fibrin being activated, forming the clot. One of the first things that happens, though, is we get what's called a platelet plug forming. You have a hole, and you're like, oh my gosh, what are we going to do? If you have a hole in a pipe, you might like jam something in it, like jam a rag in it or something like that, just to kind of slow it down. The platelets do that originally. So when that blood vessel breaks, platelets that are floating around all the time will start sticking to the wall of the blood vessel, sticking to each other, and helping to form what's called a platelet plug. But that's not a permanent solution, or it's not a long-term solution. We have to involve the fibrin to really reinforce it, like we said. Okay, so what we're going to do is we'll talk a little bit more about those plasma constituents, but we'll spend a lot of the lecture talking about the cellular elements. We'll talk about where they come from, how long they live, what they do when they're living, and then where do they go when they go to die. Okay? So we'll hit each of those in turn. But this table has a lot of information in it, a lot of core information that would be good in terms of studying for the exam. So we'll add in some additional detail, like we said, like, okay, how are the 5 different white blood cells different? But there's a lot of good information in here that you should definitely learn, and then if you want to make flashcards or whatever, could be helpful. Okay, so information about the blood itself. So you guys know a fair bit about blood. You've seen it from time to time, hopefully not too often. But we know that the coloration that we have is red, and it's not red and blue like some of the models would suggest. It's red and bright red, dark red. It's just red, okay? So that kind of depends on how well oxygenated it is. If it is well oxygenated, the red blood cells have a lot of oxygen bound to it. It tends to be a brighter red. And then if it has less oxygen, it tends to be a darker red. So we mentioned how there are 3 opportunities for extra credit during the semester. One of them is donating blood. How many people have donated blood already? Not like this semester, but you've done it before sometime. Okay? So you've seen, if you did it or somebody else did, you've probably seen that when they do that, they actually do the needle into a vein. So they do it into a vein to an artery. If they do it into an artery, they're not doing it right. So arteries, high pressure, pulsing because of their relative location to the heart. So if you hit an artery, it's like shooting across the room. It may not be that bad, but it's got some pressure behind it. You don't want that, outward pressure. So the vein, low pressure, low to slower flow, etc. And then the blood drains from the vein, and it goes into the bag, and when you see it, it's not blue, right? It's still red, darker red, but still red. So we'll talk about where that coloration comes from in a little bit. pH of the blood is tightly controlled. It's between 7.35 and 7.45. So we know that that scale, or we might remember that that scale goes from 0 to 14, the pH scale, right? In the middle of that is 7, that's neutral. On either side of that, it's going to be either acidic or basic, acidic or alkaline. So 7.35 to 7.45 is on one side of that neutral center. So is that slightly acidic or slightly basic? Yeah, 50-50 shot, even if you don't remember, right? So that is slightly alkaline, slightly basic. The small numbers are going to be more acidic. The smaller the number, right? Even up to 0. There's a 0 as well. That's extremely acidic. And then the larger numbers are going to be basic or alkaline. So that is slightly alkaline. It is very tightly controlled. If you get outside of that range of the blood, you go either into acidosis or alkalosis. And since the blood serves all these different systems of the body, now those become too acidic or too basic and they start malfunctioning. Bless you. So we have a chapter later on in the semester where we'll talk about fluid balance and pH balance and things like that, and we'll talk about that a bit more. But pH in the body we know is pretty important, and the blood is really tightly controlled. Temperature is about 38 degrees. It kind of depends on where it is. We usually measure temperature towards the surface of the body, right? So like in the ear, the forehead, or under the tongue, or something like that. But the core body temperature is actually a bit warmer than that. So you keep talking about that 98.6 degrees or whatever, 38 degrees Celsius. It varies, right? So core temperature is going to be a little bit warmer, and even between people it's going to vary. Like I don't usually have 98.6 degrees. I'm usually a little bit cooler than that. In fact, through time, they've kind of discovered that most people are a little bit cooler than that. But, so it also, though, it helps to maintain homeostasis in terms of temperature, because the blood itself is warmed and cooled in various ways. But think about if you get cold, right? So these days you might get cold every once in a while. What happens, right? Usually at the periphery is where you get cold, right? Your hands get cold, your nose gets cold, your feet gets cold, something like that. And that has to do with a dynamic process where your blood vessels, through a feedback mechanism, are either constricting or dilating. They're controlling the blood flow to these peripheral areas. So if it's getting colder, we want to keep it more towards the core and make sure all these critical organs are staying warm. So we're going to constrict out the periphery. We're going to keep it in a little bit further, right? So you might get a little bit pale too, right? So your hands or your feet might get a little pale, your skin might get a little pale, etc. On the other side of the coin, if you're exercising or it's hot out or both, you have too much heat. We still need to maintain that internal temperature. We don't want it to get too high. You need to get rid of some heat. And so you'll often dilate the blood vessels, as we talked about when you did integumentary system. Dilate the blood vessels. Now that warm blood's near the surface, and we're radiating that heat off instead, right? So that's why you kind of get that flushed appearance instead. So it definitely helps with regulating blood temperature and body temperature as well. Okay. In terms of how much, on average, you have about 5 liters of blood. Now that also is going to vary based on body size, etc. But it's about 5 liters. So 2 2-liter bottles and then a 1-liter bottle. That's about it. And we try to maintain that as well. So not only the percentage and the makeup of it, but the total volume also. All right. So what we found in even talking about the different cells and talking about the things in the plasma, etc., is that the blood has a variety of different functions, right? So it's connective, like we said. It helps to regulate things and distribute them to different parts of the body. And then we know that there are some protective functions. Protective is in, I don't want to lose all this blood, so we're protecting our system and our body, but also protective in terms of being on the alert for anything that might be infecting us, etc. So for these slides that we see here, like I said, these text slides, I'm not going to go through these because we already really talked about them when we were looking at that table. Okay? So that'll get us to bump through here a little bit faster. This is where we were talking about before in terms of the plasma. I did reorganize this slide just a little bit, just to write in and write out those 3 plasma proteins. It's nothing different than what we saw on the table, but I just wrote out those 3 plasma proteins and put in their functions, because you want to make sure that you know those, right? So for things like this, you could get it as a multiple choice question. For instance, it says something about, okay, so what is the substance in the blood that helps to maintain osmotic balance? And then it lists these 3 different plasma proteins, and then it lists, I don't know, sodium as well. Well, it could be involved. But anyway, you could be able to pick it out, or like we said, if we have a essay question, and it says, list 5 different components or 5 different substances we find in the blood, you could use these particular plasma proteins or any of these other substances. Okay. So what I would like to go to then is go over to our formed elements instead. So I'll start with red blood cells, we'll go into white blood cells, finish up with platelets, and platelets will lead us into talking about blood clotting and blood typing. So we'll probably get to that instead on Wednesday, a week from today, but that's kind of where we're going to be going. So like we saw in the slides and in the pictures, etc., we call these formed elements or cellular components or cellular elements. Don't let that throw you off. We're just talking about the different types of cells that we have, or cell fragments that we have in the blood. We saw that only the white blood cells are the complete cells, red blood cells, and the platelets are definitely not whole cells in various ways. And it depends on kind of what their role is and what they're doing at the time as to how long they survive. There are certain blood cells that may last you almost your entire life. So we'll talk about later on with our immune system how it has memory to it and how it can remember and protect you from certain pathogens throughout your lifetime, right? So how many people in here have had chickenpox? A few, right? So others, maybe you haven't had it, but maybe you've had, now they have the chickenpox vaccine, right? So somehow, whether you get it naturally or whether you get the vaccine, you're supposed to only be able to get it once, right? There's something about your body that remembers that you were exposed to it and protects you from it in the future. And usually that aspect of memory comes from certain types of white blood cells and their products that will then you have to last a lifetime, right? If your cellular memory only lasts a year, then what happens 5 years down the road if you get exposed to chickenpox again, right? So those cells can last a lifetime, but there are other cellular elements like the platelets that may only last a few days, right? They're just shreds of cells, so it's not surprising they're not going to last too long, right? Okay. Now, as we go through, yeah, Jason.
Can cells like pass on that cellular memory? Like let's say they, like when they regenerate, so like let's say that cell dies, but they pass it on to the next cell?
That's a good question. So there's definitely communication between cells. We'll talk about different types of chemical messengers or chemicals where they can stimulate each other and kind of, if you will, let each other know what's going on or bring them on alert, etc. In terms of passing on the memory itself, it's more, if you want to consider it this way, it's more like cellular genetics. You know what I mean? So, for instance, if we talk about genetics and it's like in a certain cell line or in a certain breed of dog or something like that, there's certain ones that are bred to be, like in my morning class, we had one of the students, her name was Mindy Doodle. Yes, Mindy Doodle. So her name was Mindy, the word that went along with her was Doodle, because she has a golden doodle. That's the dog, right? That's a new dog. I didn't have those dogs when I was growing up. Everything changes. But anyway, like the Labradoodles and the Golden Doodles, they're a blend, if you will, of Poodle and Labrador, or Poodle and Golden Retriever. One key aspect about them is that they're kind of hypoallergenic. They don't shed a lot, and so if you have somebody who has allergies, and one of her kids has allergies, then this is a way that you can have a dog, but without getting them all crazy with the pet hair and things like that. So, point being is that it's through the production of generations where those heritable traits are passed on, that those characteristics kind of are then carried on. And in cells as well, you can have a cell that has become activated, etc., because it's been exposed to a particular pathogen, and then that cell can divide, and the additional cells that are made are clones of that original cell. So in that way, they will have the same abilities as that first one that was able to fight off that pathogen. Yeah. So it's pretty cool. Okay. So what we're going to be doing, like I said, is we'll talk about where these different cells come from, how long they live, what do they do while they're living, and then where do they go when they die. And we're going to start with the erythrocytes, or the red blood cells. Now I can tell you, spoiler alert, that basically all of our different cellular elements come from the bone marrow. Okay? So you guys learned about bone marrow in the skeletal system last semester. You actually learned that there are two types of bone marrow, right? What are the two types of bone marrow? Red and yellow, right? So the yellow bone marrow is actually fat, right? So when somebody says that they're big boned or something like that, and that's the reason that they're, it contributes, right? So in fact, that's where they store some of their energy, is you can store it in the bones as well. But that energy storage occurs not only in bone. That energy storage, they found that yellow marrow can actually be changed into red marrow, but it's the red marrow that's the main one that's important in terms of what's called hematopoiesis, the production of blood cells. Whether they be red or white or platelets, it comes from the bone marrow. Now I say that, and there's always exceptions to the rules. We'll talk about that later on. But for the most part, all of our different blood cells come from the red bone marrow. Okay? All right, so erythrocytes, to be sure, we recognize these already. We know that they are kind of unique in their shape, right? They're biconcave discs as opposed to being round. They are anucleate. They don't have a nucleus to them. And they have essentially no organelles. As they go into the bloodstream and they mature, they give up their nucleus. They give up a lot of their organelles like mitochondria, etc. Because their main role is to transport oxygen via the molecule that we have is hemoglobin. So hemoglobin is a protein, a 4-part protein, that is in the plasma membrane of these cells, and that's where the oxygen is transported. So their main role is basically like a bag of oxygen. And so if that's your one role, you try to streamline, if you will. I mean, that's kind of anthropomorphizing it. But functionally, these things are just transporting oxygen around. So why do we need a nucleus, right? We just need them to transport oxygen. Why do we need other organelles? We don't need them to reproduce, just transport oxygen. When they wear out, we'll just replace them. Okay, so it's kind of a disposable system. All right. So hemoglobin is a critical protein that we find in red blood cells. There is another protein called spectrin that gives them some flexibility and allows them to change shape. These cells, as they're going around, they need to go near almost every cell in the body, right? Because the blood's going to be delivering all that stuff. Oxygen, nutrients, water, things like that. So we have to have a distribution system that goes just about everywhere. But as we'll look at the blood vessels go from very few, very big ones, like the branches of a tree, to more numerous but smaller and smaller branches, down to the capillaries. And the capillaries are so small that many of them are small enough so that only one cell, one blood cell at a time can fit through. They have to go single file, and sometimes they even have to flex, like bend, in order to squeeze through these capillaries to do their job and deliver oxygen. So this spectrin protein helps them to retain this kind of flexibility so that as they squeeze through there, they can then kind of open up again and continue on the way through the veins. So we've seen them. Kind of look like donuts, except it's not a hole in the center. It's just kind of squinched in the center like that. And they think that that shape is adaptive because it gives them more surface area to hold more hemoglobin rather than just a circle like that. So on the left we see what it looks like in the microscope. We can see some of those white blood cells as well. And on the right is a scanning electron micrograph. So rather than using light, it uses electrons. You get a really detailed image. And so you're seeing a cross-section of a small vein. All those red are the red blood cells, and all those white are some white blood cells. So you can see how they look like a normal cell instead. Okay, so we talked about their biconcave shape and how that is a good way to carry even more hemoglobin. In fact, they're about 97% hemoglobin. And any ATP that they need for cellular activities, they generate anaerobically using glycolysis. Because the deal is this, right? The whole idea is that they pick up oxygen near the lungs, they hold on to it and distribute it around the body, and then they release it. They deliver it to the tissues that need it. So imagine, if you will, that sounds a little bit like how some staff at a restaurant might work, right? So the wait staff of the restaurant, their whole role is to take your order. Hey, what do you need? Oh, you need that? Okay, I'm going to go get it. So they go to the kitchen, they go get it, and then they transport what you need back to you. But imagine, if you will, that you had some wait staff that after they load up their tray and the plates of the kitchen, they decide that they're hungry and they're going to eat off of your plate, right? Like DoorDash or something like that. No. I mean, like one of those other ones. Sorry. So anyway, so if they take some of what they're supposed to deliver, that's not a good delivery system, right? So it's the same thing in terms of red blood cells. If they're supposed to be delivering oxygen, if they had mitochondria, they would be consuming oxygen in order to produce their ATP aerobically, because that's what it takes. So they get rid of those mitochondria, and any ATP that they may need, they do so using glycolysis instead. So again, totally streamlined for their particular function of carrying oxygen and not using it, but delivering it. So this is what the hemoglobin molecule looks like. It has 4 different components to it. It has these 4 proteins, the 2 alpha ones and the 2 beta ones that you see on the left-hand side. And then, in addition to that, you'll notice that there is a little green, almost like spaceship in there as well. So we can see it right through here. There's 1, and there's 4 of them, right? So all 4 of those are the heme groups. These are basically the binding pockets where the oxygen go. So for every hemoglobin molecule, it can bind 4 oxygen. And then there are thousands upon thousands of these hemoglobin molecules in one red blood cell. And then, like we said, in like a cubic millimeter of blood, there's like 5 to 6 million of them. So there are literally trillions of them, if you will, going around, delivering exponentially, not exponentially, but lots and lots of more molecules of oxygen in them. So in those heme groups, at the core of it, we have iron. So you have to have iron to make functional hemoglobin. It creates a temporary binding to oxygen and then can carry it around. Okay? If you don't have iron, then you don't have functional hemoglobin, you can't do your job. So you may be anemic. So that's why one of the different types of what's called anemia is called iron deficiency anemia, or commonly dietary anemia. So if you don't have enough iron in your diet, Iron's usually associated with eating meat because meat's made of muscle. Muscle has a really intense blood supply to it because it's an active tissue. And so if you eat meat, then you're getting some of the blood in there, so you're getting the iron that your body needs as well. Certainly you can do it as a vegetarian, as a vegan or whatever, but you have to be a little bit more conscious of it. It's like what types of foods are high iron foods, etc. Anyway, you have to have that iron to make functional hemoglobin. And if you don't, you may become anemic. And there's other ways of becoming anemic, but iron is really critical there. Okay, so if we have hemoglobin binding the oxygen, it actually gets a special name. We call it oxyhemoglobin. That's pretty straightforward, right? If we've given up some of that oxygen and it's become oxygen poor, it's called deoxyhemoglobin. That's pretty straightforward too. That's the difference between, again, that bright red and that darker red blood. Because when you have oxygen binding in those pockets, in the heme groups, just like if you have something metal that's outside and it gets exposed to the air and the water and stuff like that, it's going to rust through time, right? So you have that oxidation, if you will, of the iron that's in the metal, and it gives it that red color. So same thing here. When the oxygen is bound temporarily to the iron in those heme groups, it's a brighter red. So we have oxyhemoglobin, we have deoxyhemoglobin. It just depends on how much has been delivered to the tissues, or are we still holding it in the hemoglobin unit itself. Interestingly enough, we always talk about how hemoglobin can hold oxygen, but it can bind other things as well. So for instance, we said each hemoglobin molecule can hold 4 oxygen molecules. So as it's going around, it's going to pick up oxygen at the lungs, and then it's going to go down to the tissues and deliver it, right? So as it goes down there, it's going to maybe drop off 1 or 2 molecules. When it does that, it basically has a free hand. It's been carrying these things, and it goes, oh, okay, you can have an oxygen. Now I have a free hand. So it is possible for hemoglobin to bind other things as well. It can. We know that from aerobic respiration, we need oxygen. But one of the byproducts is it produces carbon dioxide. So when it delivers the oxygen, it can say, oh, you got some garbage there? Let me grab that for you. So hemoglobin can actually bind and carry carbon dioxide as well, help to take it back up to the lungs so that we can exhale and get it out of the body. So in that case, we call it the carbaminohemoglobin instead. So it can bind a few different things. Interestingly enough, it can actually bind hydrogen ions as well as carbon monoxide as well. The hydrogen ions in a fluid, that's the definition of an acid, right? And we know we have to regulate pH in the body. So if things are getting too acidic, if we grab that hydrogen out of the fluid and we bind it to the hemoglobin, then it becomes less acidic. So the hemoglobin in the red blood cells can also help in terms of pH balance with the body. Carbon monoxide, right? Carbon dioxide, what's the equation for carbon dioxide? CO2, right? Carbon monoxide, CO, just has 1 oxygen instead. So carbon monoxide is what? It's a byproduct when you are burning, if you will, combustible materials. If you have an engine running or something like that, right? The exhaust will have carbon monoxide in it. It's very dangerous because carbon monoxide will actually, what we would call, preferentially bind to hemoglobin. That means if hemoglobin, if you will, like personifying it, if hemoglobin comes up and there's an oxygen molecule there and there's a carbon monoxide molecule there, it will choose the carbon monoxide molecule over the oxygen. So even if there's a little bit of carbon monoxide that is accumulating in the air around you, your hemoglobin is going to start grabbing that instead of grabbing oxygen, even though there's oxygen there. That's why it's important that, right, if you have something running, that you have it properly ventilated. That's why it's also usually a building requirement that they have a carbon monoxide detector somewhere in the house, usually located somewhere near the garage where you're going to get those types of fumes. Because you can't smell it, you can end up, right, passing out because now all of your hemoglobin, not all of it, a lot of it is holding carbon monoxide. Now your different tissues are becoming oxygen deprived. And so for your brain and things like that, you might pass out. So, in any case, we see that hemoglobin in the red blood cells can bind a variety of things in addition to oxygen. Okay, so let's talk about the hemoglobin and the erythrocytes together. How do we get these erythrocytes originally? So the erythrocytes are generated via a process that's called erythropoiesis. Now, what you see up there is hematopoiesis. That's basically the creation of any of our type of blood cells. Red blood cells, white blood cells, platelets, etc. We know that erythro means red, that prefix. So if we're talking specifically about the generation of red blood cells, we call it erythropoiesis instead. And where does it occur? Like we said, it occurs in the red bone marrow. Typically in our bigger bones, where we have more spongy bone, because that's where the red marrow is, that's where you're going to get a lot of that. So for instance, in the pelvic bones, right, they have some thick parts to them, right? So we have the big ilia that send out, we have the ischium that's towards the back. Those thicker areas have a lot of spongy bone in it. You get the expanded areas of long bones, like the head of the femur and those places. Even the sternum has some good spongy bone in the center. So all of that red marrow is hematopoietic tissue, and then we're going to get our red blood cells coming from there. It takes about a couple weeks or so to generate these red blood cells from, starting with like a generic stem cell, and then developing it into a mature red blood cell and releasing it into the circulation. And that's why, for instance, if you choose for extra credit to donate blood this semester, you can't be like there every week or 2 weeks. You're like, here, take some more. I want to try to get some more extra credit. And again, that's why we only let you do it once for extra credit. Because it takes a while for your body to recreate it. So you are going to be anemic after you get rid of those red blood cells when you actually donate blood like that. If you donate plasma, then you can actually regenerate that a little bit faster. You can donate that faster because plasma, we said, is the fluid component instead. So it's very easy if you increase your fluid intake, etc., then it's much easier to replace that. Although it's always funny where it talks about donating plasma, right? You're not really donating the plasma because they're paying you for the plasma. That's different. But it's a good way to, pay the rent, whatever.